You know, our virtual field trips have been a real hit, not only, you know, in the state of Washington, but around the country, across the country and around the world. Folks have been tuning in and they just love learning about the programs that are going on in our national parks. Today, we're going to learn some about bats. And this is a topic that Tara Chestnut specializes in, and Michael Hansen is getting her master's degree now in the field. And so with that, I'm going to dive in. Again, I'm Lori Ward. I'm CEO at Washington's National Park Fund. We're glad you're with us all today. We've had another really strong year at the fund in spite of COVID, and staff is healthy, board is healthy. A lot of our donors are calling in and showing their love and support for Mount Rainier, North Cascades, and Olympic National Parks. I know a number of you are out there today, and so special thanks to everyone. Before we begin, just a few housekeeping tips. This is a 45-minute program. The presentation will go right up until about 12:30, 12:35, and I'll jump back in. We'll answer some questions and answers. You're encouraged to put your questions throughout the presentation in the Q&A box down at the bottom, and we'll get to those at the end of the program. So with that, also, excuse me, please enter or please know that we do have closed captioning enabled, which you can gain access to along the bottom of the screen. If you have any questions about that after today, reach out to us and we'll assist. So with that, are your packs packed? Do you have your lunch ready to go? And let's board the bus and head on up to Mount Rainier National Park. First, a bit about our guest today. Michael Hansen, thank you for joining us, Michael, has worked in wildlife biology since graduating from the University of Oregon in 2015. She's been with the National Park Service at Mount Rainier for the past 3 years, and her first 2 years were focused on monitoring the northern spotted owl. What a great opportunity. And this past spring, she transitioned into the bat program at both Mount Rainier and North Cascades National Parks. She's also working towards a master's degree in wildlife science at OSU. Her graduate research is focused on investigating the ecology of bats during their winter hibernation and fall breeding seasons. So we'll be hearing some about that today, I'm sure. And second, we have with us Dr. Tara Chestnut. Tara and I have been friends for a very long time. She's an ecologist at Mount Rainier National Park, where she manages the wildlife program. She's one of the folks who's been involved with the marmots and the fishers, and most recently the sighting of the wolverine up at the park. Tara has a PhD in environmental sciences with a focus on disease ecology from Oregon State University. She also has 2 master's degrees focusing in aquatic science from Portland State University and environmental policy from the Evergreen State College. Tara has attended the Evergreen State College for her bachelor's degree in non-game wildlife ecology. With that, I'm going to turn it over to you, Tara, you, Michael, and say thank you for joining us and take it from here.
Thank you so much, Lori. And I'm going to turn off my video in a second just because my internet is a little bit slow, but I just wanted the participants to have a face to the name. So hi, and I'm going to stop my video and share my screen. Well, good afternoon, and thank you for joining us today. Michael and I have a fun presentation for you filled with lots of learning that's going to take us on a journey of science topics, including biodiversity, taxonomy, ways of knowing, and conservation work in Washington's national parks. We chose these topics to meet learning standards for K-12 science education in Washington State for our online learners. And peppered throughout the presentation, you'll notice social media hashtags so you can follow up to learn more about the topics and the scientists behind the work. So I hope you're ready to buckle up and get to learning. Before we get started talking about bats, I thought we'd begin with some learning for our participants who are in school and some relearning for our participants who have been out of school for a while. One of the ways that scientists study and understand the world around us is by classifying things. For living things like plants and animals, we classify them based on the shared derived traits that they have. In the example on the right, we see how different types of animals are classified from kingdom all the way down to phylum based on their shared traits. Let's start out with a few questions to help us think about the diversity of living things in the world. And so we'll have a poll that you can participate in, asking how many total living species do you think live on Earth?
All right, so if you want to pick one of these options for how many total species occur in the world, we've got 500,000, we've got 1.2 million, 4 million, 8.7 million, or 10 million. Which one do you think is the correct answer? Let's see, it looks like most of you said 10 million, 56%, followed by 8.7 million, and then 4 million, and then just 3% of you said less than 4 million.
All right, well, the answer to this question is a little bit tricky because scientists don't really know. We know how many species have been described to date, which is about 1.2 million. But we're constantly making new discoveries and advancing our ways of knowing. And so 8.7 million species are estimated to occur on Earth. Because of scientific advances in the last 50 or so years, scientists are describing around 18,000 new species per year, and many of them are microorganisms like fungi and bacteria. However, we have a lot of work to do in the future, and I hope this sense of discovery inspires the younger scientists who are here today. So for our next question, we have another poll.
Not seeing that poll yet. There we go. How many total animals occur in the world? Do you think that that's 250,000, 500,000, 1.1 million, 2.1 million, or 3.1 million? All right, it looks like the majority of you thought it was 3.1 million with 34%, followed by 2.1 and 1.1, and then looks like about 20% of you said less than 1.1.
All right, well, the total number of animal species that have been described to date are 1.1 million. But interestingly, or perhaps for some unexpectedly, invertebrates like flies and bees and iceworms, nudibranchs and squid account for 97% of the animal diversity in the world. Vertebrates like fish, amphibians, reptiles, birds, and mammals make up only 3% of the world's animal diversity. And so for our final question, how many mammals do you think are currently described?
All right, so how many mammal species occur in the world? We've got 2,400, 3,400, 4,400, 5,400, or 6,400. What do you all think? All right, it looks like 33% of you went big again with 6,400. It looks like roughly 20 to 30% of you sort of interspersed among the rest of them, and just 7% said 2,400.
Scientists have described approximately 6,400 species of living mammals. 20 to 25% of all mammals on Earth are bats. The only mammal taxa that are more abundant are rodents. 14 species of bats occur in Washington state, and 11 species occur in Western Washington. They include moth specialists like this Townsend's big-eared bat, beetle specialists like this hoary bat, and fly, midge, and mosquito specialists like this little brown bat. Biodiversity is essential to human well-being, but people have been reducing biodiversity throughout human history. Natural resources provide habitat and connectivity for a wide number of species which benefit humans and include medicine, landscapes that are resilient to change, food security, and the retention of cultural resources. Extinction has always been a feature of life on Earth, but the domination of global ecosystems by humans has caused a sharp rise in the rate of extinctions to far above pre-human levels. Here we see the cumulative extinction of mammals over the past 60,000 years, and recent trends in the percent of threatened bird, mammal, and amphibian species assessed using criteria established by the International Union for the Conservation of Nature, or the IUCN. Our knowledge of current threats suggests that the rate of extinction could soon rise to at least 5 times higher than it has been in the past. We know that all species are connected to others through ecological interactions. Extinctions therefore reverberate through ecosystems, as do extirpations of local populations and declines in abundance. So how do we know biodiversity losses are occurring? Long-term population monitoring programs provide vital information in species trends over time. Well-known examples of early community science projects that some of you may have even participated in include the Breeding Bird Survey and the Christmas Bird Count, as well as more recent projects like iNaturalist and eBird, which allow everyone to contribute their observations from anywhere in the world. Museum collections show us how to quantify, or museum collections allow us to quantify range changes by comparing historic museum records and modern observations. This example from the Grinnell Resurvey Project in California National Parks provides evidence that the montane shrew, a very small mammal, has shifted its range to higher elevations in the last 100 years. Hunting and trapping records have been used to qualitatively estimate the commonness of fur bearers and introduced predators. And traditional ecological knowledge or indigenous knowledge of natural resources provides information about species' natural history, abundance, distribution, and phenological shifts. An example of how globalization is impacting humans and other species is the spread of invasive species and emerging infectious diseases. Animals are transported all over the world for many reasons. The pet trade is one way diseases can be accidentally introduced to new places, and there are current efforts in the United States proposed to reduce this threat by requiring disease screening of imported pets and restricting or eliminating importation of high-risk species. Animals can also accidentally be introduced by transportation. Brown tree snakes were introduced to Guam from Australia and Indonesia with devastating effects to birds and other fauna. Transoceanic shipping has long been a vector for the movement of species on ships, as we've seen with global rodent introductions. Frogs from Oregon and Washington have been transported to Alaska on Christmas tree shipments. And bats have been detected in containers, including one from Southern California that was discovered in Hawaii on a ship bound for New Zealand. Bats elicit a variety of responses from people: awe, wonder, curiosity, fear. Understanding the important role bats play in our environment and communicating that importance to others can help reduce the negative feelings some people feel towards bats. For example, bats provide critical ecosystem services by consuming crop and agricultural pests that is valued at more than $3.7 billion per year in the United States alone. In 2006, people in New York State began reporting bat activity in winter when they should be hibernating. At many of these sites, bats were observed dead or dying near their winter hibernation sites, and more than 95% of some bat colonies perished due to a disease called white nose syndrome. After intensive study, scientists described a fungal skin pathogen that was not previously known. White nose syndrome appears to have been imported from Europe, where bats developed a resistance to the disease. We don't know how white nose was introduced to North America, but it was very likely from international transport. White nose only affects bats during hibernation when they shut down their vital systems, including their immune system, so they can survive the winter when food is scarce. It does not affect humans, pets, or other animals. In spring of 2016, white nose syndrome was detected in King County, Washington, over 1,000 miles from the nearest known location. This was an unexpected jump, but National Park Service scientists in the region were prepared. What is the National Park Service role in responding to emerging infectious diseases like white nose syndrome? Well, it's the mission of the National Park Service to preserve, unimpaired, the natural and cultural resources for the enjoyment, education, and inspiration of this and future generations. To fulfill this mission requires employing and collaborating with people of diverse backgrounds and expertise. The National Park Service is organized into regions. The Pacific West region includes Washington, Oregon, Idaho, Nevada, California, and the Pacific Islands. And within the Pacific West region, parks are organized into networks for the purposes of the inventory and monitoring program. While the National Park Service has a small footprint in terms of acreage, the agency brings much to the table with regard to white nose response and bat monitoring. The red star indicates the area where white nose syndrome was detected in King County, which, as you can see, is just a short distance from Mount Rainier National Park, just about 30 miles from the Carbon River drainage. After the 2016 discovery of white nose syndrome in Washington state, National Park Service ecologists and managers formed the Pacific West Region White Nose Syndrome Response Team to coordinate white nose response among parks within the region and with other government agencies. Our team includes people with prior experience working with bats, scientists and veterinarians with expertise in white nose syndrome and disease ecology, and communication specialists. Our team quickly assembled and implemented a regional white nose response strategy to help inform, unify, and guide parks in planning their response efforts that was consistent with the U.S. Fish and Wildlife Service National White Nose Response Plan and the Washington Department of Fish and Wildlife White Nose Syndrome Response Plan as well. The strategy identified the major areas of activities and need: training and resources, bat monitoring, white nose surveillance, and communication, with all of these activities anchored in collaboration and coordination. Prior to the 2016 white nose detection, we only had one biologist in the North Coast Cascades Network who had experience working with bats, which means we needed to get folks up to speed really fast. Bats are different than most other animals that I've worked with, and they're really, really challenging to study because they require specialized equipment, software programs, and rabies shots, none of which are cheap. And the learning curve is really steep. With the support of the Pacific West region, we increased our capacity and now have more than 15 additional biologists trained in bat handling and identification, acoustic monitoring and call processing, and we acquired the equipment needed to implement acoustic bat monitoring, as well as direct and passive white nose surveillance. And we have fun doing it, except for maybe this Mexican free-tailed bat that I think looks a little bit like Eeyore. So in 2016, we started out by conducting a data mining project to synthesize information about the species that were documented in the park to identify any known hibernacula or roost sites, and also synthesize past research and monitoring activities. We also reached out to maintenance staff and integrated pest management coordinators to identify suspected bat roosts in buildings. And we did this particularly because female bats have a very high site fidelity to their maternal colonies and will return to the same site year after year. Okay, so question time. This is our last poll. How long do you think a little brown bat can live?
All right, so how long can a little brown bat live? Do you think it's going to be a little over a year, over 5 years, over 10 years, over 15 years, or more than 20 years? All right, looks like the majority of you went with just about 5 years. That was 43% of you, with 37% going for 10 years, and just 12% of you said over 20.
That is a really, really great guess, especially based on how long animals of similar size live. This little brown bat that we're banding and measuring here, it weighs about 5 grams, which is about the same weight as a nickel. And but they can live for more than 20 years. They have a remarkable life history strategy, and they're very, very long-lived. Acoustic monitoring is one of the best tools we have to understand bat activity throughout the year and changes that may be a result of white nose syndrome spread or other regional stressors like impacts from wind farms. National parks are our most protected areas, and if we see changes in bat activity in parks, our coordinated monitoring program can help to inform conservation at broader landscape scales. Our elevation acoustic monitoring study, which was designed by Dr. Rebecca McCaffrey at the USGS and coordinated by both her and Michael Hansen, is gathering baseline information about bat activity in our 3 mountain parks prior to the spread of white nose syndrome. This work will inform how we design a long-term bat monitoring program in the future. We contribute to the NA BAT program by sampling priority grids and contributing those data to a continental scale program that evaluates the status of bat species in the United States and Canada. We work with the Northwest Bat Hub, housed in the Human and Ecosystems Resiliency and Sustainability Lab at Oregon State University, where we're initiating a continuous acoustic monitoring program at all of the parks in the North Coast Cascades network. This will help us learn more about what normal bat activity looks like at each of the different parks, so that we can potentially detect changes in activity should white nose syndrome or other stressors be impacting those populations. And Michael is also conducting opportunistic sampling to detect autumn bat activity that will inform her master's research on bat natural history and movements after they leave maternal colonies, because we want to learn where they go to hibernate so we can target those areas for white nose surveillance. Acoustic monitoring isn't the only tool we have to detect bats on the landscape. With potential roosts and hibernacula options in trees, cliffs, and talus that are as vast as our mountains, we work with the assistance of rogue detection teams using trained dogs to track where bats go in forested, cliff, and talus landscapes to supplement acoustic monitoring and inform more targeted white nose syndrome surveillance efforts. And so we don't currently know where bats in Washington's national parks hibernate. So the next best time to conduct surveillance is in May and June when they return to spring colonies. Bats that survive the disease heal quickly and clear the infection by early June. We conduct direct white nose surveillance at 6 bat colonies in 4 national parks by capturing and swabbing bats on their skin. Clinical signs of the disease include this orange fluorescence when a UV light is shown onto their skin. If we observe this, we collect a tissue biopsy to confirm infection. We collaborate with Dr. Tal Levy and Dr. Jenny Urbina at Oregon State University for sample analysis. And if they detect a positive, we submit a duplicate sample to the USGS National Wildlife Health Center for additional confirmation. We also conduct passive surveillance at bat colonies by collecting guano throughout the season. We collect guano, also known as scat, fecal pellets, or simply poop. Scat provides a remarkable amount of information about the animal that deposited it. We can conduct disease screening to know if a pathogen may be present in the population. We can identify the species, the individual, and also what it ate. The methods are constantly improving and rapidly advancing, so it's a really exciting time to be a poop scientist. For example, Dr. Urbina led an experiment and determined that DNA from the fungus that causes white nose syndrome can be detected in guano in very low concentrations for up to 70 days. This is important because it informs disease surveillance sampling and response strategies in national parks and other places. Unlike direct disease surveillance, which can only be done in winter and early spring, we learn that we can conduct non-invasive surveillance by collecting guano for early detection of the disease at new sites and identify the species that are likely infected. This reduces the stress caused by animal handling and also increases the pool of people who can collect samples because you don't need expensive rabies shots or experience with animal handling to contribute to surveillance efforts. Our national parks attract over 300 million visitors per year, providing the National Park Service with the opportunity to educate the public about important issues like bats and white nose syndrome. The National Park Service has been a leader in conducting inventories and long-term monitoring of natural resources with high-quality science and state-of-the-art data management as its hallmarks. We have numerous scientists and park managers conducting bat research, monitoring, and habitat management. We have an annual internal competitive source of white nose syndrome funding related to education, management, resource protection, surveillance, and monitoring. And the National Park Service has all of these opportunities and capacities combined with the spirit of collaboration, particularly with an emphasis on connected conservation, which stresses the importance of collaboration with other land managers and partners to support healthy ecosystems at the landscape scale. So what can you do to help? Well, when you're visiting parks or traveling away from home, check your tent flies, canopies, and awnings before you leave to make sure that you're not accidentally taking wildlife with you, bats, insects, or other animals. Before leaving your campsite, clean seeds off of your vehicle, your tent, your clothing, shoes, and other gear to avoid transporting them to a new area. And be sure to wash your vehicle and gear frequently, and being sure to remove all mud. If you're a cave enthusiast or visit places where white nose syndrome could occur, decontaminate your equipment to ensure that you're not accidentally spreading the disease to new locations. A new decontamination protocol was just published, and you can find it on whitenosesyndrome.org. At home, you can improve the habitat around your property by leaving standing snags where it's safe to do so, putting up a bat house, and planting wildlife-friendly landscapes. Even a few native plants in pots on your apartment patio or deck can help. You can also get involved by volunteering to help with monitoring or restoration projects in parks, and you can donate. The Washington National Park funding from the Washington National Park Fund helped 9 interns get the experience they needed to launch their careers as wildlife biologists by providing funding for housing and vehicles for 3 interns. It also allowed us to pivot our internships this year to remote situations so that we could be COVID safe. If you're interested in learning more about bats and white nose syndrome in national parks, I encourage you to go to our website and check out this video. And with that, Michael and I can take your questions.
Wow, Tara, that is so fascinating. There's so many things, and thank you both, Tara and Michael, for the program very much. Folks asked questions in advance, and I want to pose a few of those to you. What are baby bats called? That's from one of our younger audience members. What are baby bats called?
Oh, that's a wonderful question. Baby bats are called pups.
Thank you. That was, I agree, it was a good question. Somebody's wondering, you mentioned beetle specialist. Can you explain that a little bit?
I'm sorry, will you say that again?
Yeah.
Or...
Someone asked about beetle specialists, and we saw that on the screen, a couple different specialists. What does that mean? Does that mean that what does that mean? I won't even take a shot at that.
Yeah, that's a great question. So insects and invertebrates are the most diverse of all of the animals, and beetles are everywhere. And they're also an important forest pest. We've seen recent die-offs in some of our true firs that were caused by beetles, and also in our pines. And so bats like hoary bats and silver-haired bats, they eat primarily beetles. And because of that, because of the very hard shell that they have, and because they're so maneuverable in their flight, they have one of the strongest bite forces of any animal. And so the force that their jaw is required to catch and then consume a beetle is incredibly powerful. And there's a mammologist at the University of Washington, Charlene Santana, who studies that exact science.
That's fascinating. I'm going to ask Michael real quickly, and then we're going to go to the Q&A box. Thanks, folks, for the thoughtful questions here. Michael, it sounds like Oregon State University specializes in this. Is that accurate?
Specializes in what? I'm sorry.
In this, that's okay. In the study of bats, do they have a special program earmarked for that?
Well, my program is actually relatively new through my advisor, but I know that a number of other professors at Oregon State University have focused on bat research in the past.
That's great. We appreciate that. What are the animal predators of bats? I throw that out to either of you. The animal predators of bats.
I'm going to try to turn on my video, and if my face freezes, I apologize, and I'll turn it off. Okay. But just to join the group again. So there are lots of predators of bats. Peregrine falcons are a famous predator of bats. There are several owl species that eat bats. And if mammals, particularly carnivores, are lucky enough to find hibernacula, they could certainly consume a lot of bats. But at some of Michael has a wonderful story about watching falcons and other hawks eating bats as they're coming out of their cave.
Oh yeah, we've had a couple of, well, here at the park also, we had an incident where some interns called in a panic because there was a barred owl that was attacking their emergence count. They were watching the bat box. This owl was just...
Michael's freezing up just a little bit. Yeah.
Earth in Borneo. And it's one of the largest bat colonies in the world. And when those come out, I can't remember how many millions of bats come out, but basically it's just a feast for hawks. And there's a long snake of bats coming out of the cave and just hawks crisscrossing through that stream and picking them off. That's pretty impressive.
I bet it was. I bet it was. Thank you. One of our friends from Whidbey Island has a brown bat colony on her property, and she believes that this is a nursery site, but she notices that they are still around right now. They seem very healthy, but is it possible for her to send a guano sample for testing somewhere?
So the Washington Department of Fish and Wildlife is the manager outside of national park lands, and you can search their website on how to report both bat colonies, because I think that they'd be very interested in monitoring, and if you have the capacity to do emergence counts, that would be amazing, and also screening for white nose syndrome.
Great, thank you. And anyone, when we're done here, you can reach out to us and we can hook you up with Tara and Michael. So just know that. How many types of, well, what does the fungus do to the bat? How does it affect their internal systems? Are there antibiotics that treat it if it's caught early?
That's a really great question. And so the fungus that causes white nose syndrome is a skin infection. And so it's not a systemic infection that gets into their blood or their organs. And one of the main ways that we think it causes mortality in bats is that when they're hibernating, they store up as much food as they possibly can. So they pack on fat so that they can survive the winter. And when they hibernate, they shut down most of their core functions, and so they lower their breathing and respiration. They shut down their kidneys, so they're not urinating as frequently, and they're not eating, and so they're using that fat store to help them survive for in some cases many months. And the infection on their skin is really irritating and itchy, and it wakes them up, and so they're using extra energy to essentially scratch themselves and to try to groom the fungus off of their skin, and by using that energy, it causes them to starve.
Wow. Let's see, I've heard
and so
climate change.
there is interest in just for the second part of the question, there is interest in investigating treatment options. The problem is with a lot of just widespread diseases, it's really difficult to combat them once they're introduced, which is why it's really important for us to have policies in place, like related to the international pet trade and shipping, where we're training people to recognize when plants and animals are arriving to new places, like the fact that workers in Hawaii recognized this tiny little bat that they knew what to search for, prevented an introduction of a bat to New Zealand, where it could have brought other diseases. There is some promising technology that is being investigated at some of the large cave parks, where they're investigating whether we can use UV light to decontaminate sites. And in Texas, there are some really interesting experiments with mines that have a lot of bat colonies in them, where they're using a product that is basically it's Docalax, which is a laxative, a glycerin-based laxative product, to coat the walls of the mine so that it makes a barrier that the fungus can't get through. And then there's also some treatments that Tal Levy and others that he's working with are investigating that could also be promising. But right now, there's not a universal treatment for white nose syndrome.
That is fascinating, Tara. Thank you. We have time for just a few more questions, some good questions here. You mentioned there are 14 bat species in Washington. Which are the most common and which are the rarest? Do you have a sense of that?
Do you want to take that question, Michael?
Well, as far as the ones that we encounter, we end up with a lot of little brown bats and western long-eared bats. But we're looking at bats that behave in a certain way. We have some other larger high-flying bats that aren't necessarily caught by our survey methods, like silver-haired bats and hoary bats. And I'm actually not sure how they compare to the overall population of bats in Washington.
Thank you. I've heard climate change has increased fungal activity. Is this a contributing factor to the white nose syndrome?
We don't know at this point how large scale global stressors like climate change are influencing the spread of white nose syndrome, and so it's really difficult to answer that question with any real evidence. I think that disease ecology and fungal pathogens in particular, that was the focus of my PhD research. And so I joke that like fungal diseases are my jam. I enjoy them or understanding them a lot. And there are some examples where there is good evidence that fungal pathogens and the increase or spread are linked to climate change, particularly like in corals. But we don't know. I think it's too soon because white nose syndrome has only been around since 2006 in North America. So I think it's just too soon to really know or answer those questions.
Last question before we ask you to put the website back up for the video. And again, folks, everyone who's with us, this will be posted to our website tomorrow, the entire program. Last question, are bats especially susceptible to rabies or do we just happen to catch and test bats more often than other animals? Good question.
That's a great question. There has been recent research that evaluated the proportion of bats, wild bats that are infected with rabies, and it's less than 1%. The tricky thing about rabies is that it's certain death, and so there is no treatment, there is no cure. If you are exposed, which actually happened to me, you have to get a post-exposure rabies series, which can protect you as long as you get that series in time. But that's very expensive. It costs about $1,000 to get the pre-exposure rabies series, and I was billed $20,000 for the post-exposure series. But what's important about rabies is that it's a virus that changes the behavior of its host. And so animals that are infected lose their fear. They bite. Their behavior is really different. And so anytime we have close encounters with wildlife, including bats or raccoons or even cats, where their behavior is a little bit off, I think it's reasonable to expect rabies, which is why it's important if it's a pet to keep them up to date on their shots. And if you're uncertain, you can either call the public health department and ask them what to do. In some cases, they'll recommend that you actually trap the animal and bring it in for testing, or your doctor may recommend that you get the rabies series because it is just a terrible disease. But I don't think that there's evidence that that bats are infected any more than other animals. I think, but in Washington, bats are the most common animal that we detect rabies in. Second are house cats.
Can you pull up the video link, the website that you showed there?
You bet.
And one last question. What are all the species of pine cone? Does that make sense to you, that question?
All the species of pine cones?
Mm-hmm.
Um,
You can pass on that one.
well, I am, well, I'm not a botanist, but I am familiar with the pines that we have here in our parks. We've got white bark pine, we've got oh goodness. I'm going to stop my video so I can show this.
Thank you. That's
White
great
bark.
right there.
Thanks.
Yeah, Lanny, that's what I'm pretty sure that's what you were looking for. I want to toss out, if anyone has additional questions, be in touch with us. And this again will be posted tomorrow. All of our webinars get shown day after, and they're all, every webinar, or I'm sorry, virtual field trip that we've done is posted on our website at WNPF.org. And I just want to say thank you to the two of you, Tara and Michael, for your program today. It's fascinating to me. I learned so many little nuggets, and they always stick with me, these programs. Tara, I look forward to Sharon London, who's behind the scenes here. She and I look forward to your program in about 2 weeks on Wednesday, November 26th, when you'll be back to talk about carnivores at Mount Rainier. And then a few weeks later, Patty Happy over at Olympic National Park will be with us again to talk about the marmot research that they're doing every year. So if you're big fans of the marmots, tune in. Thank you all for joining us. If you're interested in supporting this program, go to our website and donate now. We're grateful to all of you. Again, Tara and Michael, thank you very much. Have a good afternoon.
Thank you, Maria and Sharon, and for everyone that participated.
Thank you. Have a great day, everybody.
You guys too. Take care.